All right, good morning, everyone. So today we're going to go ahead and talk about DNA, structured DNA. We're probably also going to do a little bit on protein synthesis. These are two of the, especially protein synthesis, a little bit more difficult content to understand. So I definitely wanted to make a video on this. All right, so let's go ahead and start out with DNA. Now, DNA, you guys have learned about before. We're just going to kind of go into a little bit more detail. So as a reminder, your first module, we went over nucleic acids. So which was one of the macromolecules. DNA and RNA are both nucleic acids. That's what the Na stands for in DNA and RNA. All right, so just as a reminder, it stores genetic information in the form of all these different nucleotides and the order that these nucleotides are in. That's just different chemicals. All right, so first of all, the physical structure, you've got the sugar phosphate backbone right here. On the outside or the rungs of this twisted ladder, and then you've got your nucleotides in the middle. So these are four different nucleotides, four different chemicals represented by the letters A, T, C, G. Now, you have like adenine, for example, would be A. So if I am talking about A, T, C, and G, those are specific nucleotide specific chemicals. Right, let's move on. Here's a 3D image of what this looks like. So again, it's just looks like a ladder, but twisted. That's why it's called a twisted ladder, or a double helix. That's why it's called that. Now, something to keep in mind as you're looking at the structure, if you notice, the red is always with the green on the rungs of the ladder. Purple is always with yellow. Okay, you're never going to see a red with a yellow or a green with a purple. It's always going to be red and green, purple and yellow. Okay, that's because those letters that we talked about earlier, those nucleotides, A, T, C, G, they're always paired up the same exact way. Okay, that's called base pairing rules. So A and T are always going to be paired together. C and G are always going to be paired together. Okay, it's always going to be like that. So that'll make it easier when you do some DNA replication here in a minute when I show you that. Okay, and then there's RNA. Okay, so RNA, we also talked about, it's also nucleic acid. RNA is a single strand. So just like in this picture, there's only one strand. So DNA stores your genetic information. And then, so RNA actually builds, it does the building. So RNA uses the information on DNA to build a protein. And that's basically the whole process of protein synthesis. It's how RNA actually builds proteins from the information on DNA. Okay, and then another difference in RNA, RNA has the nucleotide U instead of T, uracil instead of thymine. So also keep that in mind. And then RNA can be anywhere in the cell. DNA is generally, it's just going to be in the nucleus. It can't leave the nucleus. Okay, and then here's a 3D image of RNA. All right, so on to DNA replication. So we talked about the cell cycle, and the first part of the cell cycle is interphase, which consists of G1, S, and then G2. So this is the S phase of the cell cycle that we're talking about in just a little bit more detail. So again, S stands for synthesis. So synthesize means you're making, specifically you're making DNA. So again, so how this works, so you have that double helix, that twisted ladder, it's going to unwind and then it's just going to split in half. So kind of like this picture, there's like a zipper there, just splitting in half. Okay. And then it's going to take the old strand. So you have two old strands. So each old strand is going to use as a template to make a new strand. And it does all this with enzyme. So there's one enzyme that goes down and it splits it in half. And then there's another enzyme here that adds the other strand of the new strand of DNA. So this is what we call a semi-conservative. So half of the DNA is new and then half of the DNA is old. So you end up with two new strands, each with one new, well, half of it new, half of it old, to summarize that. All right, so I had mentioned base pairing rules before just a couple minutes ago. So again, you know, you have A is always going to be with T, C is always going to be with G. So if you look at just, if this was half of the strand, of DNA, if there's an A there, there's always going to be a T next to it. If there's a C there, it's always going to be G next to it, or vice versa. If there's a G, there's going to be a C, so on and so forth. A slight difference when you're going to RNA. We'll talk about that in another section, but just kind of keep this in the back of your head. So normally in DNA, A goes to T, A matches up with T. When you're going to RNA, A goes to U because there's no T, so A goes to U. That'll make sense in a minute when we do some practice. So the complementing strand, it might be phrased like this on your exam, of ATC. So all you do, so if this is half of the DNA sequence, half of a strand of DNA, you're writing what would be attached to it. So this is just your base pairing rules. So if there's an A, like there is here, okay, you would have a T. So then there's a T here, so T is always attached to A, and then C is always a G. So this would be T, A, G. The other half of that DNA strand would be T, A, G. Okay, here's another one. If you want, pause it, make sure you can do it yourself. So GTA, always in the class, there was always one student that would go, oh, Grand Theft Auto. So GTA, if you flip this, G goes to C, T goes to A, and then A goes to T. All right, and I'm going to go ahead and go through this practice question 2. So here's 4 different segments of DNA. So when you're looking at this is half of a DNA strand, this is the other half it's attached to. And these are just 4 different segments. So to find the error in the DNA, you're going to go through and see if you can find one of these that where an A is not matched with a T or a C is not matched with a G. So T is with A is with T, G is with C, G is with C, C is with G. This segment is completely fine. GC, GC, TA, GC, AT. This one is completely fine. GCAT. Okay, there's an error. A does not go with T. I mean, C. A goes with T. So segment 3 is one error. We can go ahead and jump to segment 4, CGAT, AT, TA, GC. That's completely fine. So the error is on segment 3 only. All right, I'm going to go ahead and close this out, and then I'm going to open up the protein synthesis one. All right, so when we talked about RNA and briefly DNA and DNA replication, so when the DNA replicates, that's the first step of, it's basically the first step of protein synthesis. You have to have the DNA replicate. So you have to have more DNA. So anyways, protein synthesis is, like I said, it's basically the process where proteins are made from the information on DNA. So the simplest way to put this is you have DNA, that information is transferred to RNA, and then that's basically turned into a protein. That's the simplest way to put it. Okay, so now to go into a little bit more detail. So step 1 is what we call transcription. All transcription means is you're going from DNA to RNA. So kind of similar to what we did with DNA replication, where, if you had an A, you put a T. If you had a T, you put an A. C, you put a G. You do the same exact thing, except when you're going to RNA, A is going to turn to U instead of turning to T. That's the only difference. Everything else is the same. Okay, so that's transcription. Okay, so during protein synthesis, there's 3 different types of RNA that's going to be created that's used for this process. These are just the functions of it. So they kind of tell you what it is in the name M. M stands for messenger. So it's carrying. R stands for ribosomal, carries part of the ribosomes. If you remember, you learned what ribosomes do. Ribosomes make proteins. So it all kind of ties together. This shows the importance of how everything kind of ties together. And then transfer RNA. It helps decode the mRNA. mRNA actually is what ultimately gets turned into the protein itself. So when it's talking about translated in the form of a protein, that's just a fancy way of saying turned into a protein. Okay. All right, so step 2 is translation. So step 1, you have DNA to RNA. Step 2, RNA, that RNA that we made is going to get turned into or translated into a protein, specifically from the mRNA. And so it has to turn into amino acids first, multiple amino acids. Again, first module stuff we learned about before. Amino acids make up a protein. It's the monomer of a protein. So there's technically a little step in between here where the RNA is turned into multiple amino acids, and that amino acids, depending on which amino acids you have, turns into a specific protein. Okay, and this occurs in the cytoplasm and ultimately in the ribosomes. Again, ribosomes make proteins. Make sure you know that. Proteins, you should think amino acids. So if you can make all these little connections, it makes your life a lot easier. All right, and kind of on the same line, just some terms you'll see. So when transcription occurs, you have mRNA, which is what we could call a codon. Think of a codon. It's just 3 of these letters together. Codon, it's a code. So every 3 letters is going to represent a specific amino acid. You'll be given a chart on your exam and any of the assignments you do of, which, so GCU might be alanine, for example. I don't know if it is, but you'll be given a chart that says all that. And then tRNA is what we call the anticodon. So so this is not what gets translated. The mRNA is what gets translated. All right, so we'll do this question together. So again, if you want to pause it, see if you can do it yourself. Otherwise, I'm just going to go ahead and tell you how to get the answer real quick. So how many amino acids are coded in this segment? So don't worry too much about the 5 and the 3. They just kind of mark the beginning and the end of this segment of DNA. So don't get too caught up on that. So to figure this out, amino acids, one amino acid is from one codon. And from our previous slide, one codon is 3 nucleotides, so 3 letters. So every 3 letters is one amino acid. So you've got 1, 2, 3, 4. So your answer is 4. Okay, so this table on the right is an example of one of those codon tables you might get. So every 3 letters. So if you read this particular one from the inside out, you might be given another one in a different type of table. But either way, every 3 letters. So let's say you want to look at C, G, U. So C is the first letter, G is the second letter, and then U. So that'll give you this amino acid right here. Okay. And then this whole slide is about genetic code. So this codon table is the same for every single organism. So that code I just gave you, GCU or whatever I said, doesn't matter if we're talking about a fish or a bee or a human, it's always going to code for adenine. Okay. And this leads to some really unique technology, some biotechnology that we'll talk about another day. And then, of course, this is also evidence of a common ancestor. Again, that's another topic we'll go into a little bit more detail too another day. All right, I'm not going to worry about that one. That has to do with the mutations. We're going to talk about mutations another day. But mutations, basically, just a quick rundown. So just because a mutation occurs, so a mutation is a change in DNA. So one of these letters gets changed. So just because a mutation occurs doesn't mean it's going to affect the organism. So first of all, some of the mutations don't actually, some of the DNA doesn't do anything. So even if it's mutated, it doesn't matter. You can mutate it all you want. It's basically just DNA that is just kind of junk DNA. It's just kind of there. It doesn't have a function. So mutate it all at once, it's not going to do anything. Or it could mutate it and it could become the same amino acid. For example, we were talking about CGU. So say CGU gets mutated to CGC, okay? It doesn't matter, it still codes for the same amino acid, so nothing would be different, okay? All right, so that's everything. I'll go into more detail on mutations in another talk. If you need to help doing DNA to DNA replication, if you need help going to transcription and translation, let me know, but you're just kind of flipping the letters around. And I think I made a video, I did make a video on it. It's an old assignment. They changed it this year, so you don't have to actually physically write it, but one of the old assignments, I think it's 3.05, 3.06, one of those in there. If you look at it in the video list, it'll show you more detail how to do that if you have, you know, some multiple choice questions or open-ended questions on that. All right, that's everything. You guys have a good one.